Hello, my name is Grace Inamorato, and I will be discussing case connections 2 and 4. Case 2 is concerned with the regulation of glucose uptake by normal tissues and then with cancer cells. We're shown here a glioblastoma tumor with areas of necrosis, and we are asked to discuss the normal tissue glucose uptake. And I think that it's first important to look at the hormones that are responsible for maintaining glucose homeostasis in order to understand these processes. So the major hormones that are involved in glucose homeostasis are insulin, glucagon, cortisol, and epinephrine. Insulin is an anabolic hormone that is released by the beta isolate cells of the pancreas, and levels are elevated during the fed state. Insulin has tyrosine kinase-linked receptors that fuse with plasma membrane to expose active transporters. Autophosphorylation of the receptors causes the tyrosine residues to be exposed for binding. Transporters are internalized when insulin levels fall, reducing glucose uptake. Insulin promotes the direct glucose uptake by membrane transporters, promotes indirect glucose uptake by increasing phosphorylation of glucose to glucose 6-phosphatase, and promotes conversion of glucose stores. Glucagon is a catabolic hormone that is elevated in the fasted state. It allows for the breakdown of stored fuels to be used as energy. Glucagon initiates glycogenolysis and produces glucose through gluconeogenesis. Glucagon is a guanine-binding protein, or G-protein coupled receptor hormone that triggers the cyclic AMP pathway. When the hormone is bound, conformational changes in the transmembrane domains change the plasma membrane to facilitate exchanges of GP and GTP. Cortisol is released from the adrenal cortex during the fasted state, and it diffuses into the bloodstream to bind to intracellular receptors. Cortisol enhances gluconeogenesis and is needed to supply carbon skeletons for glucagon-mediated gluconeogenesis. Lastly, we have epinephrine, which is another catabolic G-protein receptor hormone that is elevated in the fasted state, similar to glucagon. Epinephrine enhances lipolysis, glycogenolysis through alpha agonist pathway, and beta oxidation of fatty acids. Looking at the liver, glucose metabolism begins when dietary carbohydrates are directly absorbed from the intestine via the hepatic portal vein. Glucose uptake is operated through insulin-independent glucose transporters. In the liver, the GLUT2 transporter passively accepts glucose into the hepatocyte because this transporter is located primarily in the plasma membrane, also making it insulin-independent. GLUT2 facilitates a bidirectional flux of glucose in and out of the cell. Once glucose is in the cell, it is phosphorylated by hexokinase isozymes to yield glucose 6-phosphatase. Human glucokinase phosphorylates the 6-carbon of glucose with the help of ATP and is not inhibited by the product. This is demonstrated in one of the irreversible steps of glycolysis. Glycolysis oxidizes glucose to pyruvate, which is then metabolized into acetyl-CoA. The pyruvates provide a carbon skeleton for synthesis of non-essential amino acids, and acetyl-CoA will either enter the TCA cycle for complete oxidation of carbon dioxide and water, produce ATP and oxidative phosphorylation, or become precursors for fatty acid synthesis. Glucokinase resides in liver tissues, accounting for 95% of glucose phosphorylation activity, and they are controlled by glucokinase regulatory proteins, which are acted on by fructose 1-phosphate, the activation, and fructose 6-phosphate, which is the suppressor. To overcome reactions phosphorylized by hexokinase isozymes, the liver has glucose 6-phosphatase. This enzyme will dephosphorylate glucose 6-phosphate to generate 3-glucose to be released into circulation. This makes the liver the only organ that can release significant amounts of glucose into the bloodstream, making glucose available to other tissues such as the brain and skeletal muscles. Overcoming the phosphorylation by glucokinase and gluconeogenesis is important because it is one of the key regulatory measures that allow us to bypass the irreversible steps of glycolysis. The primary effect of glucagon on the liver is to promote the production and release of glucose. Glucagon initiates the use of stored energy reserves and helps maintain blood glucose concentrations during the fasted state. Glycogen is a source of reducing equivalence NADPH for fatty acid synthesis and a source of ribose for nucleotides and nucleic acid synthesis. In the fasted state, lipolysis of adipose is a pathway providing the main fuel to produce ATP, and it also produces ketone bodies from acetyl-CoA that can be used as a source of energy for peripheral tissues. Looking at the brain, glucose is the brain's main energy source, in the form of ATP through facilitated diffusion mediated across the blood-brain barrier by membrane-bound glucose transporter proteins GLUT1 and GLUT3. The brain requires continuous supply of glucose from circulation, and insulin has no effect on glucose uptake of the brain. Astrocytes take up glucose from the blood through GLUT1 receptors and metabolize it to lactate, which is then delivered to neurons. GLUT3 is of major importance for glucose transport because of low Michaelis-Menten constant values that enable it to supply continuous glucose to the neurons, even at low interstitial glucose concentrations. Hexokinase has such a high affinity towards glucose that the rate of glucose phosphorylation approximates the enzyme's maximum reaction rate. The pathway for providing glucose in the fastest state is through hepatic glycogenolysis and gluconeogenesis. Looking at skeletal muscles, unlike the liver and brain, skeletal muscle uptakes glucose in an insulin-dependent manner. Skeletal muscles only take up glucose when insulin is present or in the fed state. The glucose transporter protein responsible for skeletal muscle glucose is GLUT4. And this is a translocating transporter and are special in that these proteins promote fusion of vesicles containing the transporters with plasma membranes. And only insulin is able to do this. In working skeletal muscle, insulin is not required for uptake of glucose because exercise mobilizes the GLUT4 transporters through calcium. These transporters are always active because of the calcium ion concentrations that respond to nerve stimulations. Skeletal muscle can use glucose as fuel both aerobically, forming carbon dioxide, and anaerobically, forming lactate. During the fasted state, through lipolysis in adipose tissues, the fuels used are fatty acids. The free fatty acids are oxidized through fatty acid beta oxidation, protein catabolism, and the oxidation of ketones. Epinephrine enhances glycogenolysis via the cyclic AMP pathway. Cortisol promotes protein catabolism, and glucagon has no impact. Looking at adipose tissue, it also uses GLUT4, similar to skeletal muscle, as an insulin-dependent manner. When insulin is detected, GLUT4 is compartmentalized in intracellular membrane vesicles that fuse with the plasma membrane and deliver the transporter to its site of action. Insulin increases GLUT4 on the plasma membrane and therefore increases the uptake of glucose. When fat is broken down, it is released fatty acids into the blood. Excess glucose is stored as fatty acids through fatty acid synthesis and tag synthesis. Free fatty acids are provided through lipolysis, which is enhanced with the help of glucagon and epinephrine. Insulin controls the tag synthesis and glucose uptake through GLUT4. So, what modifications have occurred within the tumor cells? Glioblastomas are malignant grade 4 tumors, and the cells are reproducing and dividing at any given time, nourished by an ample and abnormal tumor vessel blood supply. The ability of the cancer cells to switch from oxidative metabolism to glycolysis and production of lactate, even when oxygen is plentiful, is a key characteristic. The process of increased glucose uptake and lactate produced even in the presence of oxygen is known as the Warburg effect. The aerobic glycolytic pathway that the tumors use is what brings the increased glucose flux, which then in turn maintains ATP production that tumor cells need to meet for their heightened demands. This reduces the use of the TCA cycle so that the pyruvate made in glycolysis is converted to lactate. Usually an increase in lactate would result in normal cell death. However, cancer cells can export lactate, resulting in the acidification of the immediate area. This could destroy the surrounding cells, causing the necrosis shown in the brain image. Cancer cells use aerobic glycolysis to provide macromolecules for daughter cells. Each lactate excreted from a cell breaks off 3 carbons that have been used for ATP production or for precursors for other processes. Instead, cancer cells incorporate the extra carbons into biomass for rapid cell division. This flux of macromolecules also creates a major source of reactive oxygen species, and this formation occurs when there is a suppressed pyruvate input, which then can be caused by mutations in cell electron transport. High levels of ROS promotes genetic instability in tumors, favors growth, chemotherapeutic escape, and an increase in tumor mass is also associated with the deregulated expression of GLUT1 and GLUT3. Deregulated expression occurs through a direct interaction with gene regulatory elements or by controlling cellular trafficking of the transporters to the membrane. Patients who are diagnosed with glioblastomas are at higher risk for hyperglycemia due to an increased glucose flux, and this can actually, it is believed, to stimulate tumor growth. So that completes our case 2 connection. And looking at case 4, we're presented with a 2-month-old female And with the given information, we want to find the following diagnosis for this patient. So what's important out of this? What we know. We have a 2-month-old female. She has only been given breast milk since birth. She has recently been sleeping for more than 2 hours between feedings. Laboratory results are consistent with hypoglycemia. Epinephrine administered and with an increase in blood lactate, an enlarged liver, and blood glucose levels increase after breastfeeding but cannot maintain levels within normal range during feeding. So the first intolerance that we're going to look at is hereditary fructose intolerance. And this is a condition where the body is unable to digest the sugar fructose. Fructose is found in sucrose. Fructose is a major dietary component that comes from fruit. And this is a deficiency in ADAlase B, which is a major enzyme for rate-limiting steps for fructose 1-phosphate metabolism. The results in the accumulation of fructose has pretty serious complications, and these implications include an enlarged liver, fasting hypoglycemia, being lethargic, poor feeding, fructosuria, and inhibition of glycogenolysis and gluconeogenesis from the lack of free phosphates. And this condition typically manifests during infancy at weaning when children are introduced to fruits and vegetables. So since the mother reports that she has not given the baby anything to eat other than breast milk since birth, this intolerance cannot be our diagnosis. Up next we have galactosemia, which is a disorder that affects how the body processes galactose, which makes up lactose. This is caused by a mutation or absence of the enzyme galactose 1-phosphate uridyl transferase, also known as GALT, that inhibits the body's ability to metabolize galactose. Classic galactosemia can be diagnosed any time after birth and is a primary source of energy for infants and breast milk. Affected infants usually develop feeding difficulties, are lethargic, failure to gain weight, grow as expected, jaundice, low blood sugar, liver damage, and abnormal feedings. This is not the diagnosis for our patient because she would have been tested at newborn screening and the symptoms would have presented sooner since she has been drinking breast milk since birth. So next we have glucose 6-phosphate dehydrogenase deficiency, and this is a deficiency in the glucose 6-phosphate dehydrogenase enzyme that's responsible for protecting red blood cells against the harmful effects of reactive oxygen species. And without this enzyme, the ROS levels would accumulate in the cell and cause them to break down prematurely. So the rate at which red blood cells are dying is faster than the birth of new cells. Clinical implications are almost exclusively presented in males as hemolytic anemia, jaundice, fatigue, shortness of breath, and rapid heart rate. Epinephrine would have not been administered to a patient who shows clinical implications of increased heart rate and shortness of breath. This cannot be a diagnosis for our patient since she is female and her red blood cells were not affected to our knowledge. The complications that our patient presents do not coincide with the complications presented in somebody with this deficiency. So that leaves glycogen storage disease, which is an inherited genetic disorder that causes glycogen to be improperly stored in the body due to deficiencies in glucose 6-phosphatase enzyme. We learned earlier from case 2 that glucose 6-phosphatase is found in the liver and is used to dephosphorylate glucose 6-phosphate to generate 3-glucose to be put back into circulation and later used down the line in other metabolic processes. Glycogen is primarily stored in liver or muscle tissues, greatly affecting the functioning of potentially both. Our patient is showing signs of an enlarged liver, and we can look specifically at the types of glycogen storage diseases that affect the liver. And these complications include an enlarged liver and kidneys, hypoglycemia, high levels of lactate, fats, and uric acid in blood, swollen abdomen due to enlarged liver, and possible muscle weakness. Our patient presents with an enlarged liver, hypoglycemia, and the inability to maintain blood glucose levels within a normal range during fasting, and increased levels of blood lactate. A glucagon or epinephrine challenge test can be administered in someone who is suspected to have this disease. Either hormone causes little or no increase in blood glucose concentrations, but both increase serum lactate concentration significantly. The high fasting lactate levels arrive due to failure of glycogen being degraded to free glucose molecules. So with the clinical presentations that our patient has shown, glycogen storage disease is our likely diagnosis for our patient. Thank you very much for listening to my video, and I hope that this was insightful. Thank you.